So I know people are starting to trickle in, but in the interest of time, I'd like to get this session underway. I'd like to welcome all of you online to this episode of Contagion Cultures. I'm delighted that you're able to join us and take an hour out of your day. My name is Warren Beaty, and I'm the director of the Queen's Institute for Energy and Environmental Policy. I'm the director of the School of Policy Studies here at Queen's. I'm thrilled to be able to introduce some of my colleagues who are going to be talking about wastewater and the COVID outbreak. But before we get into that, I do want to take a moment to acknowledge that I'm sitting here at Queen's University on the traditional territories of the Haudenosaunee and the Anishinaabe peoples. It is my privilege to be able to work here and to study here and to learn here. I know that today in our virtual setting, you may be on many different traditional territories, many different, representing many different nations. Please do take a moment to think about that and acknowledge that. It is, as I say, a privilege to be able to live in this country and to learn from those that came before. We do have a terrific panel of presenters here today to talk to us about detecting outbreaks using wastewater-based epidemiology. We have three speakers with us. First, we have Professor Stephen Brown, who is from the Department of Chemistry and from the School of Environmental Studies here at Queen's. We have Professor Sarah Jane Payne, who's from the Department of Civil Engineering in the Faculty of Engineering and Applied Science. And finally, we have Dr. Geof from the Beaty Water Research Center. And so welcome to all three members of our panel. We're delighted that you're able to join us and delighted that you can share your thoughts. One last thing before we get underway, for those of you online who would like to ask questions, there's a Q&A function at the bottom of your screen. If you move your cursor around, it will pop up and you can post questions in that Q&A box or you can post questions through the chat function. I'll be monitoring both as we go. So to get us underway, I believe I'm going to hand over to Sarah Jane. So please take it away.
Great, thanks, Dr. Maybe. Yes, so thank you for the introduction and also for inviting us here today to the Contagion Cultures series. My colleagues, Dr. Brown, Dr. Hall, Sophie, and I brainstormed for a title befitting of Contagion Cultures, and we came up with Flushed Intelligence. Because today we're going to discuss what we can learn about ourselves as a community through wastewater surveillance, and specifically how wastewater surveillance can be used as a tool in this pandemic. Next slide. So today we're going to discuss wastewater surveillance, our current work in the pandemic, detecting the SARS-CoV-2 virus, the efforts here at Queen's in Ontario as well as globally. We're also going to talk about wastewater-based epidemiology as a rapidly evolving field of study and future efforts, and also some of the unique challenges in rural and remote populations. And with that, I'll hand it over to my colleague, Dr. Brown.
Thank you. So this project sort of evolved within or among a group of us at the Beaty Water Research Center as we sort of talked about what people were doing in response to the pandemic, like people do. And at the same time, we were contacted by a number of people, including Utilities Kingston, to consider getting involved in this emerging initiative to monitor wastewater for the SARS-CoV-2 or COVID-19 virus. And I got contacted because I had been doing some work with sewage in partnership with Utilities Kingston for a number of years now. So we're what you would call regular customers. We would pull up once a week and they hand us a bottle of sewage and it's completely free. If you ever want free sewage, the City of Kingston or Utilities Kingston is your source. They're super for that. So we would pick up some sewage samples and we were looking for antibiotic-resistant bacteria, which was the topic of the day and within the World Health Organization and all the global conferences until, you know, what happened. And so we were already looking at sewage as a way of tracking the presence of antibiotic-resistant bacteria. There's a whole separate field around looking at sewage as a hotspot for the formation of antibiotic resistance, and then looking at links between that and even using wastewater testing for looking at the antibiotics themselves and talking about the use within a population. And so then, of course, the pandemic happened, and that antibiotic resistant bacteria work we were doing ground to a halt for a little while. And we started talking about other things we could get involved in, including monitoring to support the response to the pandemic. So we just showed this slide with the global case count. And of course, we've seen these trends over time. It looks like for the last couple of months that there's been a decrease, but when we look at the level we're at now, it's about the same as the level was back in the summer on a global basis. And that means there's a possibility for it to go back up again. There are still lots of cases out there, lots of potential sources of further infection. So then we also have the emergence of variants of concern. We'll say a bit more of those in a few minutes. And so now we're in kind of a race between vaccination and propagation of more contagious variants. And we'll see, are we going to have another increase in the near future or not? And there's lots of speculation on that. So when we look at the Canadian numbers, we see a similar kind of story where we had an initial case count with the outbreak back in the spring. We had a larger outbreak around December and January, and we're just sort of coming down from that now, but levels seem to have, or the trend seems to have leveled off. And again, the question about what's going to happen next. And so this plot came from the Public Health Agency of Canada just a few days ago, and we can zoom in on sort of the present and the near future. And this is based on modeling, including the variants of concern, but then also looking at, are we going to keep opening things the way we've been doing for the last few weeks across the country? Are we going to put some measures back in place because we're worried about the variants of concern? And we see some signs of that now as some areas have gone back into lockdown, if you like, even just this week, or are we really going to get serious and close everything down and reach that blue curve on the bottom? And so we really don't know which way things are going to go in the next few months. And, are we going to be in one of the worst case scenarios? Or are we going to be in a best case scenario or somewhere in between? And it's really difficult to predict that. And so that's where the whole wastewater-based epidemiological approach comes in. And so a thing maybe people don't think of is that when folks have this particular virus and are sitting on the toilet, for instance, then they're actually shedding virus. And we know that because we can take COVID-19 patients and analyze their stool samples, and sure enough, the SARS-CoV-2 virus is found there. And that means every time they flush the toilet, some of the virus is going out into the distribution or into the sewage system. Now, for us, the sewage system is just this fabulous collection system. It's picking up little samples from all the people in the community and bringing them down and combining them and mixing them up, and then providing one composite sample right at the inlet to the sewage plant. And so that's a really interesting place to go and test and get a result that you can say corresponds to the vast population within the community without doing person-by-person testing. There are some key advantages there too. When we talk about people getting individually tested, there's usually something that triggers testing. Most often it's because someone has symptoms or they have some kind of close contact or other high-risk event. And so there's a question about all the pre-symptomatic and asymptomatic carriers out there, and can we get a handle on what the number of those sorts of individuals would be? And so they'll be picked up through the sewage because, again, there's evidence that even if you're pre-symptomatic or asymptomatic, we start to see the virus in the stool samples. This is also really potentially a more efficient way of screening an entire community. So you get one sampling point and you're getting this mixed sample from the entire group. And of course, this is attractive on a global sense too, in places like low income or low resource settings, where you can monitor an entire community with just a few samples at a much lower cost than doing individual testing at the same scale. So that's an introduction. And so a number of groups have gotten into doing this, and indeed there's been some positive results. So this slide mentions a result that came out of the University of Arizona back in the fall, where in September, when all the students were moving in, they went around and started getting sewage samples from the major residence buildings. And from a couple of buildings, they identified samples that were positive for the virus. Now, these were students who had been at least screened for symptoms, so they were definitely asymptomatic or pre-symptomatic carriers, but they were able to lock down a couple of buildings, go into those residences only and do a lot of testing of individuals. And sure enough, they found the cases that were there, and they feel confident that they were able to stop a residence-wide or institution-wide outbreak by using those early indications that were in the sewage samples. So that is just an illustration of what can be done with this kind of analysis. And so a number of other groups have taken this on. And here we just show some results that came out of Rob Delatola's group at the University of Ottawa. And they've really been the Canadian leaders in doing this kind of work. So here's an outline of the method. So you get a sewage sample. It can be the liquid sewage. In some cases, they will take a sludge sample from just inside the sewage plant where the sewage comes in. The test that's being done at the end is the same qPCR test that's done for the clinical testing. So it should be clear, we're not using some kind of rapid test or something with maybe lower accuracy and so on. We use the exact same test that's used for the clinical samples. The challenge is that sewage samples need quite a bit more treatment or different treatment than the clinical samples need. Sewage samples can contain a lot more background chemical interference for the test. And so we get worried about things like inhibition of the PCR detection system by different contaminants that might be in there. The other thing that we point out is the detection limit we require to do this work is much lower than the clinical test. And again, it comes back to that idea that the sewage system is collecting stool samples from a wide number of people, bringing them down and collecting and mixing them into one place. And that means if we only have a few cases in the community, that all the healthy people's poop is diluting out the poop from the people who have the illness. And that means the virus is going to be at much lower levels in the sewage than we would find in the nasal swab samples from those same positive people. So we actually have to have a lower detection limit in this case than you would have in the clinical cases. And so that's been a bit of a challenge. And a lot of the work that we do in setting up the method is about trying to get the detection limit as low as possible so we can see these very trace levels of virus in the sewage. The other thing that we have to take into account is that sewage samples are variable. So anytime there's a rainstorm or this time of year when we have a warm day and there's a lot of melt and runoff, that's going to dilute the sewage with non-fecal material. And we have to have a correction for that as well, because it will end up affecting the level of viral particles that we detect. So there's lots of fecal indicator parameters that we can monitor, and that's indeed part of the investigation of the method fine-tuning that's still going on. So if we look at the Ottawa group, they have now published some of their work. And so in this paper, they showed that they detected sewage signal or virus signal rather in the sewage 2 days before case counts increased in the community. And we'll look at that data on the next slide. They have another publication where they looked at the levels that they measured over a longer period of time in the sewage, and they can indeed correlate it back to the number of cases in the community. So these are the kinds of things you would hope to be getting out of the results from analyzing the sewage. And indeed, we're starting to see proof that this can work. So this slide shows some of the data from the Ottawa group. And what we see is this solid line, which is the viral level in the sewage. And then we see the shading in behind, which in the top plot is showing the number of new cases being reported per day. And in the bottom plot is monitoring the percent positivity in the tests. And the lower plot may correct for any effects due to changing in the number of tests done in a particular community day to day. And so you can see that the data is quite similar, but there are some subtle differences. And we're still exploring which of those is the best one, and it may depend on which questions you're asking as to which data point from the community perspective you want to try to correlate the sewage data with. These results, they call these normalized viral copies, and in this case, they are correcting for sewage dilution using something called the pepper mild mottle virus. So this is actually a vegetable virus that's on peppers and similar vegetables. So when we eat them, there's a virus on them. The virus is totally harmless to us humans. It's a plant virus. Some of it goes through us and turns into a signature signal in our poop. And we can use that to correct the results. And indeed, these results show much better correlation after that correction. And so what you can see with this data is back here in the middle of July. This is where they say, look, the signal in the sewage went up at least a couple of days before the case count in the community went up. So there's evidence of doing that. Now, there was another increase in the community in September. And that didn't show up right away in the sewage. It showed up a little bit later. And so we're still learning about different contexts and situations and other parameters that have to be included in these studies to really understand the relationship between the two. So there's still some science to be done in determining those relationships before we can come down with an ironclad equation that connects the levels in the sewage to some particular parameter about the cases in the community. In this case, one of the theories is that in September, a whole bunch of people went back to school or work and had to go get tests. And therefore, a whole bunch of additional cases were caught by testing without necessarily the cases in the community having been increasing over the same time. So there are logical ways of explaining these, but more research is needed to really define it. Now, towards the end of December, Ottawa had another significant spike as part of the most recent wave, as we've been calling it, that hit most of the province. And in this case, the sewage signal came up a number of days early. And so the guys in Professor Delatola's group were pretty convinced the community case count was about to take off, and then sure enough, it did. So that does illustrate that there's some real potential for having some advanced warning about changes in the community by doing this kind of analysis. That gets everybody excited about wanting to have this kind of information available, not all by itself, but along with all of the other community health information that we can have. The group in Ottawa are now moving into the sewer shed more, so they're moving closer to certain locations like long-term care homes and so on, and taking sewage samples that are more local. And this group was just in the Ottawa papers about a week ago. They've started screening for these variants of concern, and indeed they found this the so-called UK variant in one of the sewage samples from one community where it was known there was an outbreak. So again, we can, as additional questions come up about locations of outbreaks or the variants of concern, the wastewater-based monitoring can keep up with those questions as well. So in Kingston, we're now collecting samples twice a week from the 2 wastewater plants here in town and analyzing those. This is part of the Provincial Wastewater Surveillance Initiative that's been set up by the MECP. We're also part of a national coalition hosted by the Canadian Water Network and supported by the Public Health Agency of Canada. And NML is the National Microbiology Lab in Winnipeg that's doing a lot of the fundamental analysis, method development for this. And we're in the process of expanding so that we'll also be monitoring samples from Loyalist Township, the plant out in Amherstview, City of Cornwall, and we're going to start taking samples in the Kingston sewer shed as well. So we can ask additional questions and try to answer them with our data as we go forward. Now, last thing I'll mention is we know Kingston's levels have been very low. So when we get into these discussions nationally, we're like the boring cousins. You know, everyone's talking about their outbreak and how high their levels were in their sewage samples this week. And we sit around and say, yeah, our levels are still low. We detected a baseline level in a few samples, but nothing exciting. So we sit around kind of hoping there's an outbreak, and then we look at each other and say, no, actually, we don't want an outbreak. But if there was one, we would at this point be ready for it. We do point out that there's been some research done to determine if you optimize your method entirely, how low can you get your detection limit? And in this particular paper that came out a few weeks ago, showed that they could get down to about 4.4 active cases per 100,000 population. So we're now working to try to make sure we're down at that level, which means, in Kingston that we kind of oscillate a little bit above and a little below that over the last couple of weeks. So again, we'll be in a position to start seeing signal when the levels, when the case count goes up to any degree above where we are now. And we're also analyzing some archive samples. So we did start collecting samples back in June before we had all of the equipment in place to do the analysis. And so now we're working our way through those, and we'll see, can we detect the fairly small cluster of cases that happened back in July, often called the nail salon outbreak, and then the much larger cluster of cases that we did get back in December. And we'll, again, use that to prove to ourselves that the optimized method really can get down to that 4 cases per 100,000 level, and we should be able to see some signals around these peak areas that are noticeably above the baseline. So this is being done in conjunction with public health. They're going to install an attachment on their COVID information dashboard that you'll be able to click on and see at least a weekly update of what the sewage results have been doing, depending on what the results are. And so that's going to be available as part of the public information provided around COVID as we move forward. So watch for further notices about that in the month of March. And you'll know that our project's moving ahead and we're on the case. So with that, I will turn it back over to Sarah Jane to talk a little bit more about wastewater-based epidemiology in general.
Thank you. Yeah, so we also want to talk about wastewater-based epidemiology as a tool, not just for our current COVID-19 pandemic. So one of the questions is, why is sewage so rich in information? Well, much of what we consume is not fully metabolized. So if we think about things like pharmaceuticals, for instance, we don't fully metabolize the pharmaceuticals that we take. Or in the case of SARS-CoV-2, we actually shed the virus in our stool. So if we have these unmetabolized chemicals or these identifiable metabolic products that we're excreting, or else in the case of the SARS-CoV-2 virus, we're excreting the virus itself. And if these items enter the sewage and they're stable, and we have the tools that are sensitive enough to detect them, we can actually find needles in the haystack. So if we look at the image on the right-hand side of the screen, you see this diagram of different places in the community where we're flushing toilets, entering into a sewage system, into the catchment area, and then we go to our wastewater treatment plant. So if we're sampling at the sewer shed level, so up in different neighborhoods, we can look at neighborhood level data. But if we're looking at the wastewater treatment plant, we're looking at the municipal level, so community level data. So both of these sets of data, they're not about individuals, they're about this, what's going on in the community. And this approach has been used in a number of different areas. So for instance, with opioids, which is another epidemic right now in our culture, there's been a study where they were able to look at a map of a community and identify different areas where different opioids were being used disproportionately. And they were able to develop or suggests different types of intervention strategies for those neighborhoods. So you could think about where you would want to allocate resources for counseling services or pharmacy take-back programs. So if you're able to identify where in your community you had issues, you could actually target your public health interventions. In terms of pathogens, with the polio virus in 2013, where polio is not an issue, Israel actually does routine surveillance for the wild polio virus, and they were able to detect a viral signal in their sewers, in their wastewater. And because they were able to detect this, they were able to identify specific neighborhoods where this was silently circulating. And then they were able to prioritize different neighborhoods for different interventions, including a vaccine catch-up program. So next slide. So this is an emerging field, and what we're doing right now at the Beaty Water Research Center is some of that foundational work about how do we interpret wastewater surveillance data to provide meaningful information for public health so they can do meaningful interventions. We're also asking questions about what other indicators do we need to look at to normalize sewage data, and what are the best monitoring strategies? And then what else can we learn about this experience? So if we start doing some sewage shed modeling, and in the future we look at different types of contaminants, what can tracking contaminants through a sewage shed tell us about how other contaminants can move into the environment? So the most pressing issues are, of course, the ones we face right now in 2021, where we're looking at the issue of variants of concern. And our colleagues at University of Ottawa have rapidly developed a tool to screen for the new variants that we're worried about. And this will help us anticipate future waves and of the pandemic. And also, we're in this pandemic right now, but we know it's not going to be the only pandemic. So this will help us prepare a tool to help us watch for future pandemics. And then the other question around drugs of abuse. So opioids is the other pressing public health crisis right now in Canada. So we're thinking about how we can use what we've learned so far to expand to other public health questions. And with that, I'll turn it over to Dr. Hall.
Thank you. So up until this point, we've talked about municipal systems, living in a city or a municipality that is centrally treating. But we're also, by doing that, we're missing out on a segment of the Canadian population. And the current numbers are around 14% of the population is not connected to a municipal wastewater treatment system. In Ontario, it's about 1.2 million systems that are out there. Now, there's a number of different systems when we talk about that. So if we think about it, it's just like being in the city. The wastewater is coming out of your house and has to go somewhere. Where it can go, there are a number of different recognized classes of handling that wastewater on site. Out of that, 3 of them require removal of the material at some point in its life. So we think of cesspools, septic tanks, and holding tanks out of the group. The others would be a latrine or an outhouse and a gray water disposal. But of those 3 that do require removal, they're a potential opportunity. Now, if we think about a rural setting, we're talking in general about lower population densities. So it's not as if we're going to be going to a house to sample, but we still have a population that we theoretically would like to know what's going on from a wastewater epidemiology point of view. So if we think about those different on-site treatments, some of the challenges associated with that are how often does it get removed? If you have a holding tank, you'll be removing it on a scheduled basis so that it doesn't back up and overflow. If you have a septic tank, which is providing treatment, you may have an interval of 3 to 5 years when it's pumped. Some people might pump every year. Some people might wait until there's a problem or the house changes hands to be pumped. So there are some challenges. But at some point, those three types of systems will have the wastewater and the sludge that accumulates in them and so forth removed. And generally it's a truck that shows up to the house and pumping happens. That also brings another challenge in that a hauler may service one or two houses, may service a group of houses, depending on how much is being removed and the size of the truck and so forth. We can go to the next slide, please. But generally, when the septage is removed, it's taken to either the municipal wastewater treatment facility that we've been talking about, or in some cases, there's a private wastewater treatment system of some sort that's handling the waste haulage. But it's at that point that we potentially have an opportunity to start to look at rural populations. We can take samples from the truck as they're arriving, and in partnership with the wastewater treatment facility and possibly the septage haulers, we have an opportunity to understand where they may have come from. It might be that the hauler has a specific territory that they service, so we can have an idea of where they've been. It may be that they know that they've scheduled certain areas and we can get a better sense of where the sewage is coming from. Keeping in mind, of course, confidentiality concerns and so forth. But the daily truckloads that are coming into the wastewater treatment plants present an opportunity to explore rural wastewater epidemiology and capture that part of the population that we would be currently missing with this. The other part of this puzzle is that in a rural setting, your actual source water may be a vulnerable resource as well. Sometimes when a well is contaminated, it has come from either a leaky septic system or a septic system that's not functioning as it should. And so that represents a further opportunity to connect the cycle between drinking water, wastewater, and so forth. So something we do need to be thinking about as we move forward with wastewater epidemiology is how to tap in to this 14% that is not captured with the traditional municipal systems. We'll move on to our next slide, please. So thank you all for listening. I'd like to acknowledge a number of individuals that have been critical for this to be moving forward. At the larger scale, Queen's University, the Beaty Water Research Center, the Ministry of Environment, Conservation and Parks, Utilities Kingston. And then individually, from the Beaty Water Research Center, Dr. Pascal Champagne, Nathan Mullins, Dr. Abdul Rahman Alashaf, Sophie Filleiter, and from the Kingston Health Sciences Center, Emily Moslinger, Dr. Pramit Sheff, from Ontario Public Health, Kevin McDermott, Dr. Anna Majuri, and from Utilities Kingston, Alan Lucas and Julie Runyon. Thank you. We can now open it up for any questions you may have.
That's great. And thank you so much to all of you for the presentation. I think that it's really interesting to see where the technology has gone and what is actually possible by tracking, different things within these sewage kind of lines. Now, I will remind everybody online that we do have our Q&A function open, and there's an immediate sort of comment that's cropped up, not a question, but just a comment that this is awesome. It's really helpful, particularly in the light of this pandemic. So great job there. Maybe while people online are thinking about questions, I'll pose one, and that is, how easy or hard is it to calibrate testing for different things? Like if, let's hope this doesn't happen, but another pandemic were to come along, how long does it take to get scaled up so that you can run these tests? Could we get to the point where it's kind of rapid testing? You just swap things in and out depending on what you're looking for?
Yeah, so if you think about switching things, it is quite rapid within a category. So like with viruses, I mean, what we do is we follow along behind the clinical testing. So if there's a new pandemic for something, say it's a different virus, and there's an established viral RNA test that the clinical sites are now using to detect that, and that probably happens to discover the pandemic on the first place, or at least to identify what's causing it, then yes, we can, at the back end of our process, we can drop in the same methodology for detecting the viral RNA. So it would be as fast to switch it here as it would be to get it going in the clinical lab. Now, there are some subtle questions because different kinds of viruses might attach themselves to the sewage a little bit differently, whether they're stuck in the particles or floating around. Do we need a high pH to get them to come out when we extract? Do we need a low pH to get them to come out? So there may be subtle differences between the viruses. And this is when you hear them talk about the coronavirus as an enveloped virus, for instance. If you have another virus like the flu that's not enveloped, then you may have to change your method around. Sorry, it's a different envelope. You may have to change your method around to optimize it, but otherwise, it's fairly rapid to switch from one thing to another once you're into RNA or DNA-based detection. That's an advantage.
Okay, thanks. Any other comments from the panel on that one? We do have a number of questions that have opened up. So Duncan has asked or pointed something out about rural collections, which is that it's not unusual to have a 5-year gap between collections. And that certainly means that it would be hard to do any kind of current monitoring. So is there a way to address that, or is that just something we have to deal with? Maybe I turn to Geof first, since Geof, you were talking about this, and then go around the panel.
So it's certainly a challenge, and it's part of our understanding process. And it's something that's not, it's not a part of the wastewater epidemiology at the current point. Not all septic systems are being pumped at a lower rate. I mean, if you have a holding tank, it's a lot sooner. Part of understanding it all is to partner with haulers and so forth to get a sense of what's coming. But no, that is certainly a challenge. It might be that we can use the wastewater epidemiology in rural areas for other purposes. Maybe it isn't to detect viral load changes, but we have to understand what it means, because we are getting a composite sample of different households as well as we move along. So it's part of the understanding process, just as we are with the municipal.
Okay, thanks for that, Geof. Any other comments from the panel? Sarah Jane?
Yeah, so yeah, that's a great question. And I think the other aspect to it as well is, there's communities, especially in northern Canada, where hauled sewage is actually the main form of sewage treatment. So what we can do, what Geof's proposing to do, is partly that foundational work about developing that methodology and the science behind sampling hauled sewage, so that for communities where it would be a very immediate question, we could begin to address that more quickly.
Okay, thanks for that. I think that's helpful. We'll keep on going because there's a number of questions here. I'm not sure who asked this question, but somebody's asking, what is the purpose of identifying opioids in wastewater? Anybody want to take that on?
That's good.
Yeah, sure. So, I think if you look at the other headlines that we're getting right now in Canada, it tells you about the prevalence of opioids as a drug of abuse and how deadly it is right now. And so if you're looking for opioids in wastewater, it's not individualized data, it's aggregate data collected around a community, and it would allow you to identify within a city, where do you have the most frequent use or what types are being used. And that would allow public health to target different types of interventions. So I'm not a public health person, but from what I've read, that's how it's seen as being potentially useful. So whether or not you're working with pharmacists or doctors in the area, or you're talking about different types of individual counseling that you might want to target services for, or different types of mitigation strategies, by knowing where the problem is, then you can start to target your response.
Another example for that is often you'll have a community of opioid users, and depending on the sources, you might have one day where somebody brings in a bad batch and it's laced with, say, carfentanil or some other additive that we know are spiked into these from time to time. And this would be a way of picking up, when did the sources that are coming into our community change to something that's got another ingredient in it. You could potentially pick that up through a sewage signal.
Thanks for that. Richard is asking about monitoring for influenza. Is this a type of tool that could be used in monitoring influenza and influenza outbreaks?
So let's say yes. It's a discussion we have to have with public health in the after times. Is right now our own restrictions have kept flu to a minimum, so no one's worried about checking for flu this year. But I'm sure with a number of places that have got SARS-CoV-2 monitoring in place now, they're going to want to put flu monitoring in place. And in part, it'll just be that big question about how beneficial is early detection. Now, with the flu, we don't get the contagious outbreak scenario the way we have with the COVID-19. So it'll take a more careful look at how advantageous early detection can be. But there are communities, communities do have to roll out their flu shot programs. And if they could more reasonably or more closely tailor that to the actual presence of the flu in the community, and if we could give them a week's heads up by this kind of monitoring, then I think, that would justify the cost of doing it, especially now that we've got the infrastructure in place to do the testing. To add something in and keep doing it in the long term is not going to be nearly so expensive as it was to get it started up this time.
That's important to know, and I think that could be really useful. Nishana has a pretty technical question here. Does the change in season affect the RNA? I'm not sure who would like to take that.
Anyone want it? So I think the RNA itself is not going to be affected. What will be affected is the fate of the RNA. So for a person who's shedding a certain amount of RNA, the temperature and other composition in the sewage will affect how efficiently that RNA gets to the sewage plant. And so what we might see is the calibration of our sewage level versus the community level may change with the season because of the fate of the RNA changing. I would expect to see that, in fact, whereas otherwise the RNA itself shouldn't be changing.
Okay, thanks for that. We've got a question for Geof from Anna, one of your co-investigators here. Thanks to everybody, to all of you for your presentation. The question for Geof is, with respect to the comments around rural wastewater, are we suggesting that the well is a source of infectious virus that may then result in disease in the consumer?
Oh, well, I wouldn't be making that. I think that would require more work on that. So my comment around that was more for general wastewater epidemiology, if we're looking at different contaminants and so forth. But yeah, I wouldn't be prepared to make that link.
Okay. Any other comments from the panel? I think we've covered that to some extent anyway. Another fairly technical question, and I'm not sure who's posing this one. Does a disease such as COVID-19 provoke any kind of signature in the waste other than particles of the actual virus? So would COVID stress microbes in the GI tract that could then be detected as part of a microbial signature that would complement what you're seeing with the virus. I'm not sure if it's even been investigated yet, so over to you guys. Any comments on that?
So I would say the answer is probably yes. I think this is another one of these things where if we were analyzing individuals' poop, you could probably go at this right away. In other words, you would see a change in what we call the GI microbiome. We would see that shift between people before, during, and after a SARS-CoV-2 infection. But whether we would see that in the bulk sewage from the community, there I would be skeptical. I would suggest that would take a lot of work, or it's much less likely, just because now we've got that dilution effect going on again, and a whole lot of other dynamic changes that happen to the microbial community between the poop and the sewage plant. So I don't think we would go there first until maybe after the research on the poop is done. That someone will be able to say, hey, this particular change happens that turns on this metabolite that we wouldn't normally expect to see in poop. And if you go looking for that, that could be one of these complementary indicators as suggested. But in terms of a general microbial population, I don't think we'll see it that far down the sewage line. There's some, again, maybe as we go up into the sewer shed and we get closer to the source, maybe we could start looking for things. But we'll be watching for the research on the poop first. That's where we'll find out what we could look for.
Well, that's helpful. And I think it actually partly answers Nishana's question here, which is, if the sewage is pretreated, can you still pick up traces of the virus? Or do we have to move further up the stream of the sewage, I think?
Yeah, so we currently take it at the inlet to the plant. So now, treated, we could put in quotes. The sewage has spent some time in the sewage distribution system, and absolutely that could affect the fate of the viral RNA. So that will have some impact on what we detect. So we try to get the sewage before any real treatment has happened. For that reason, we don't want it to affect the detection. Definitely by the time you get to the end of the sewage plant, whatever virus was going in, there will be less to detect. So each step of the treatment is going to remove some of the detectable virus.
Okay, thanks for that. Anna's got a follow-up, and Geof, I think we'll go to you first on this. And it relates to her previous question where she was getting at, the risk of the virus propagating in well water. Is there a risk of the virus infecting vulnerable wildlife and thus propagating in wild animals and even domestic animals? Do you see anything there that we need to be thinking about?
I think it represents a potential research opportunity for sure. Yeah, that would be my take on it.
It's a good question, and I know one of concern, particularly around pets and things that might be carrying the virus, and what might be happening with the virus inside those pets. So, spoken like a true researcher, we always see opportunities to expand our research. Amy's asking if you guys have noticed any differences in the quantity of viral RNA between the two different treatment plants here in Kingston. Any differences in the loads that you're picking up?
Go ahead, Sarah Jane.
Well, we're just skimming above the detection limit right now, and we're not 100% confident. So before we make any pronouncements, I think we'll get back to you on that, and we'll let it show up on the dashboard. But that is one of the things we're looking at is, are we going to see different levels? Because they service two different sides of the city. Are we going to see different levels? And we're also looking at how many people do each of those catchments serve. So how do we normalize that? So is there a difference? And as we get a sample within the sewage shed at a place where we think we're going to get much higher levels because we want to be near the hospital and campus, how do we reconcile that data as well? So that's a really good question about, how do we interpret the signal that we're getting? Because right now we're just looking at trends, but is there a way to get that to be more quantified so that we can start saying, there's a hot spot in our city? Yeah, great question.
Thanks for that. Anna has confirmed what we were talking about earlier with relation to influenza, that the outbreak of influenza has been hugely attenuated this year, but in other years we see lots and lots of influenza, and so this could become a useful tool, I think, is one thing to infer from that as a way to track and to keep an eye on those outbreaks. And then we have a question about new clinical detection assays and new mechanisms so that we get more and more testing in our population. Is it really important that we keep up with this wastewater detection? Is it possible that we just get to the point that we have enough testing mechanisms available through other means that we don't need this? And then the follow-up is, if the answer is yes, how do you foresee these types of tools being used in an effective and impactful way?
All right, should we take a shot at this?
Yeah.
Yeah, so it's a good question. And, by no means can we say that, wastewater-based epidemiology is the only way to answer all of these questions. There are certainly other ways. So I think if you look at how the wastewater surveillance is set up and this idea of collecting a sample from a large community, if you were out doing, and we should do the statistics on this at some point, suppose you're doing 1,000 random samples in Kingston every day, and those tests have a reasonably good low false negative rate. Are you going to get the same information that you could get from this, and that would make this unnecessary? And the answer, by the time you're up to 1,000 random tests a day, the answer might be yes. Yeah, the sewage isn't adding anything anymore. Are we currently in a situation where we expect to see 1,000 random samples a day in the long run for this? I don't think so. So now you're doing a resource trade-off. If you can get approximately the same information from the sewage monitoring, compared to the cost of 1,000 people randomly every day, then the sewage monitoring is going to win out. But it's not meant to say it's the only way to answer some of these questions, more that it may be by far the most efficient way of answering some of these questions, especially if we want to talk about doing this in the long term.
And I think also to add to that, it's the least invasive way to do this. So you don't have to knock on anyone's door. There's no issues around privacy. We're looking at the whole community. And, as new variants emerge, we can rapidly change. We can rapidly shift our tests to identify very specific signatures from the different variants of the virus, whereas a rapid yes/no test might not be able to give you that level of detail as well. So I think it's, for me, I think of this as just one of the many tools in the toolbox. It should not be the only tool, but I think it's an important tool to give us that community level data.
Okay. We've got time for a couple more questions, and there's a couple here that are sort of related. Somebody's asking, what is, or has there been a reduction in the concentration of RNA or virus detected since the vaccine has started to be distributed? There's also a question from Richard about how many people have to be shedding the virus or taking opioids or whatever in order to be able to get a detection signal. And I think those are related. Are we detecting enough right now in the system to be able to see what's going on? Any comment on that?
Yeah, I think we would start. So for the virus case, we would start by, going back to the research showing it's somewhere around 3, 4, or 5 cases per 100,000 people is currently providing enough virus for it to be detected in the sewage. And so that gives us a kind of a threshold that we would look for. Now, there's not been enough vaccination yet, or the proportion of the population vaccinated hasn't been high enough yet to say what the vaccine or what vaccination is going to do to these results. There are some really interesting questions around, when people are vaccinated, can they still get infected? Will they, they won't be ill, perhaps, but they may still have the virus. Will they be shedding it? Again, we're waiting for the initial poop analysis to answer some of those questions. And even that, it's not published yet. The research is still just emerging on that. And then we'll start to try to predict what that might do for the wastewater analysis. But even some of those questions around, are people after they're vaccinated, they're probably not getting tested anymore, but maybe they still have the virus and are shedding it. We may be able to use the sewage testing to follow that, to answer some of those questions in the post-vaccine era, if you like, and as well as to look for variants that can evade the current vaccine, can evade that immunity. We can look for those at the same time using the testing. So there's lots of questions that we can try to address where the sewage testing isn't going to be the only information for those questions, but it will certainly contribute.
There's one question here about, well, I'll read it out. The sewage infected would be from people who have been affected by COVID. What does this mean by testing, or if you are testing the entire Kingston population instead of a case study from just infected people? And I'm wondering if we've got it turned around. I think that the sewage that you're testing is from the entire Kingston population across 2 plants, right?
Yes.
So we are picking up just some infected samples from a very broad sampling. So maybe there's a little bit of confusion there. But there's a great question here from Duncan, and I think it's a good question to kind of round out this discussion on what's the effect of sewage treatment on the virus? And I think maybe what Duncan's getting at is, does sewage treatment deal with the virus, or are there chances that the virus might be getting beyond the treatment plants and back out into the environment?
Yeah, so there's been a bit of research that says the virus in poop might be infective, though the fecal oral route, as they say, is not a major one for this virus. By the time you get to the sewage plant, the infectivity of what's there is pretty low. And by the time you get to the end of treatment, the current evidence is there's no infectivity anymore. So there may still be viral particles, so RNA bound up in different particles, but it's not infectious. So when we're working with the sewage in the lab, we're more worried about other pathogens in the sewage than we are about the lab personnel catching COVID from the sewage samples. So it's not a case of the active virus being spread around in the sewage. We're just looking for the RNA signature that gives us information about the population that provided that sewage.
Thanks. I'm afraid we've come to the end of our hour. I know that there are probably more questions, and this has engendered a terrific discussion and lots of really interesting points from our viewers. So I want to thank everybody online for your contributions and for being with us today. I want to thank all 3 of our panelists, Sarah Jane, Stephen, and Geof. And I should also be thanking Sophie, I think, for her contributions with the slides. I will remind folks that this is an ongoing series, the Contagion Culture series. Next week at 4 p.m. on Tuesday, we have Kazem Termizi, who is a postdoc fellow in the Department of Global Development Studies here at Queen's, who is going to be speaking about anti-colonial readings of pandemics in South Asia, a historical perspective. So a really different take on things. I hope that you'll take some time out to join us. As always, thanks to everybody for being with us, and I hope everybody stays safe. Look forward to seeing everyone again in about a week's time. Thank you.
Thanks. Take care, everyone.
Thank you.
Have a good day.